Hello, YNP members. We have a very special guest with us today. I'll let them introduce themselves to you and yeah, take it away, Nate.
Hi, thanks, Trina. Yeah, my name is Nathaniel Ritz. I am let's see, I suppose I'm nearly done with my PhD at this point. I have all my chapters ready, and I've actually started a postdoc here at APC Microbiome Ireland. Um, as you might have guessed, I'm not Irish. I'm from New Mexico, actually, and I came here in 2018 because I had a strong interest in the microbiota gut-brain axis and learning more in this field, of which APC is one of the largest institutions in the world, really focusing on the microbiome in general and how it affects the health of the human host. So with that, I'll happily get into my presentation. I've entitled it The Microbiota Gut-Brain Axis and the Social Brain. So just a bit of background. The microbiome is very important in a number of different aspects, and I just want to kind of highlight that it is involved in many different aspects of social life, and that's not just for us humans. We see this across the animal kingdom, from honeybees who have socially horizontally transmitted microbes that can affect their social recognition through olfactory chemicals, as well as enhancing their own immune benefits from one another. And zebrafish, just below it, you can see actually differences in shoaling behavior depending on the microbiota status, whether it's been depleted with antibiotics or germ-free zebrafish. And actually in koalas, one of the key components in being able to break down eucalyptus comes from a maternally passed on microbe symbiont to the koala pups. So we see different aspects of social life being critical to health and development of all different hosts. And one of the things that we focus on here at APC is that of the host of also the human. And to do that, we, especially our group in the lab of John Krein, we focus a lot on rodent models. So we use mice and other rats to kind of make different models to test different translational parts of the microbiota gut-brain axis. So before I get too far into that part of the talk, I just wanted to also introduce that we are very much living in a microbial world where there's actually estimated to be 1.3 to 10 times more microbial cells in and on us than there are human cells that we make up us as we are. The initial estimate was a bit on the high side at tenfold, but a more recent estimate is saying that it's probably about even. Maybe we hedge a little bit more towards the microbial side. However, there is a large increase in the amount of microbial genes compared to that of the human genome. So with that, they're actually able to give us a much wider access to different genes that we are able to then use for a number of different things that help our health. So with that in mind, one of the concepts that I kind of want to introduce is our being as a hologenome. It's somewhat of a superorganism that collectively speaks of not just the host genome, but that of the host and the microbiome. Now, this is a nice paper, actually, if you want to read more into kind of the evolutionary theory behind that. But basically, as we move through time and space, our microbiome is actually capable of transmitting a lot of very helpful aspects that can kind of help us either with digestion or immune function, or even our own brain function and behavior. So through all things that we're trying to do, we're trying to look at it through the lens of evolutionary systems biology and how the microbiome can also be used to explain these different things that happen throughout life. So there's a few mechanisms that have been identified that can kind of help explain how this crosstalk between the microbiome and the gut, which harbors the vast majority of the microbes within our greater microbiome, with the highest density found in the colon. However, there are microbes throughout our body. but we really focus on the gut. So these different mechanisms, there are neurotransmitters and neuroactive metabolites that are produced by the microbiome that can actually travel through the vagus nerve, which connects the gut directly with the brain. There are also microbial metabolites and products that can either cross through into circulation or cause different cascades of functions to take place, such as short-chain fatty acids and metabolites and all kinds of microbial bioactive molecules. And then there's also the enteroendocrine system, which comprises a lot of the different hormones like glucagon, like peptide 1, peptide YY, or CCK, which kind of help our different satiety and digestion factors. And then lastly, the immune system plays a large role in how we sense what is happening in our gut and how that then can change our overall health. A lot of times when you're sick, you can notice that things start in the gut, and it can trigger a lot of different things, from brain fog to increased anxiety, or in some cases even depression. So these are some of those mechanisms. There are more when you dig into each of them, but this is kind of just a broad overview of that. All right. So to do a lot of our studies, there's been one kind of fundamental tool that we've been using a lot in the field, and that is called the germ-free model. And it's one of those kind of funny things that early in the last century, they were making comic books about Bubble Boy and different things where there's a life taken away from the environment. And that's kind of like what our model is when we use germ-free mice, where they are born and raised within a aseptic isolator that is separated from the rest of the environment and completely sterile conditions, so they don't have any access to microbes. And by using these germ-free mice, we can then compare and contrast the differences between conventional house mice, which is access to a microbiome, and that of having no microbiome presence at all. And what's really interesting is when we look at germ-free mice, we see various differences from stress hormone signaling to neuroprotection, as well as neural function. And something that I'm going to focus on a bit more throughout the talk is the behavior of these germ-free mice, which interestingly, they show a reduced anxiety-like behavior, increased cognitive deficits, changes in their sociability, and also increased stereotyped and repetitive behaviors. So what are the things that make the differences in these germ-free mice? One of them is that they show increases in myelination and myelin-related genes. Another is increases in neurogenesis in the hippocampus, as well as overall volume of the hippocampus and amygdala. There's also differences found out through neurotransmitters and synaptic plasticity. And interestingly, there's differences in dendritic growth. There's an increase in dendritic length and spine density with decreased synaptic connections and branching. In terms of microglia, the resident immune cells of the brain, we see that there's more immature microglia and less immune-related genes. And lastly, blood-brain barrier is a very important topic, and as well as barriers in general, because this kind of controls what is able to get to and from the brain, as well as there's something else which comes in handy, which is the epithelial barrier of the gut. And these different barriers largely control how the influx and efflux of different microbial transmitters, molecules, variety of different things are able to travel to and from the gut and have an overall effect on different aspects of the host. So a picture is worth 1,000 words, and in this case, you can kind of see the differences between a neuron of a typical control mouse and a germ-free animal. So the control mouse does show shorter dendritic length, but what's quite interesting is it also is much more complex with more branching, whereas the germ-free mice will show much longer neurons, but decreased branching, and this is causing numerous various differences. And then again, one of the things I mentioned was the differences in myelination. And here's a good example of how you can see that. This is the myelin layer of the cell body of the dendrite, and between control and germ-free, there's a great over-myelination, which decreases the travel of different neurotransmitters and electrical voltage function between these different cell types, which could be one of the many reasons that we see differences between the two models. We also see differences in visceral pain between germ-free and conventionally housed mice, with the germ-free being more susceptible to visceral pain in the gut compared to that of controls. And also the fear extinction recall. So using a translational model of something called fear conditioning, we see that there's an increase in how they respond to fear and their retention of it compared to that of others. Then when we look at their actual behavior socially, it's quite interesting. This is a behavior test called the 3-chamber social interaction test. Now on the left, the experimental mouse is able to either interact with another mouse or that of an empty cage. And what's really quite interesting is that the conventional mice always show a preference to interact with another social conspecific, whereas the germ-free mice lose this effect. So you can see this is just the number, this is just their paths that they take throughout the test, and the more different paths and more density of it, you can see basically their general exploration over 10 minutes. And then second part of this test is social cognition or social novelty part of the phase, where the mouse then has the option to interact with a novel mouse or the familiar mouse that it has been interacting with. And again, in normal mice, what you expect is a preference for social novelty. And that's what you see here, where there's a greater number of paths and more time spent in the novel side of the interaction chamber, whereas with germ-free mice, this effect is lost. So with this, we've actually gone and done transcriptomics of amygdala of the brain. And what we found that in this impaired sociability model, there's also altered gene expression in response to social interaction with an increase of activation of RNA splicing